Our next speaker is Temitayo from the University of Idaho. He's currently a postdoc in Brenda Murdoch's lab and has been working with Brenda and Ben Rosen and others on the bovine pangenome project. He's originally from Nigeria with degrees in computer science and engineering from Nigeria before coming to the U.S. for a postdoc. And unfortunately, he was unable to travel, so we're doing his presentation virtually. So go ahead, Idaho.
Thank you so much for the introduction. I hope you can see my pointer. I just want to be sure you can see my pointer before I proceed.
Yes.
Yep.
Okay, great. Thank you. So Temitayo Olagunju is my name. Currently a postdoc in Dr. Brenda Murdoch's lab at the University of Idaho, Department of Animal Veterinary and Food Sciences. I've been working on sheep, especially with Did you say something?
There, go ahead, Timothy. I was just adjusting the microphone here, so.
Okay, thank you. So currently working on cattle and sheep, especially with respect to Pangenome projects, and also the wider project, which is the ruminant TDT project. So really thank you for giving me this opportunity to share some thoughts with you about this topic, Pangenomes, a new era of genomics. My presentation will be guided by this outline. First, I would like to take us through what our reference genomes are, trying to highlight the shortcomings that we have with reference genomes that necessitates the use of alternative approaches, where the alternative approach in this sense is Pangenomes. So I'll try to illustrate to us the use of Pangenomes and the differences that we have seen between the use of a pangenome and a reference genome. And also, I'll go on to introduce to us or intimate us about the bovine pangenome project and some early lessons that we've learned from this bovine pangenome project. So, starting with reference genomes. Reference genomes have helped us to represent the characteristics of a species or a particular breed of a species using a single genome assembly that serves as a representative of the different breeds that you may have for that particular species. And in this case, for the use of a reference genome, research community that makes use of this resource often come together and agree to adopt a particular genome assembly, especially where you have multiple assemblies that are available for that particular species. They adopt a single reference assembly so that it becomes a base for which all analyses are made and compared. Now, most of the reference genomes that we have currently came from short reads assembly. And when I say short reads, I'm talking of first and second generation reads assemblies, where you have it's in the neighborhood of just 300 base pairs, pair reads. And using this to construct a genome assembly, you end up with a lot of gaps, a lot of missing segments, making the genome incomplete, as it were. And it is not the case that these assemblies are not useful in themselves, but when you're using other small reads to construct other genomes, then this reference assembly becomes a guide to help you to assemble the new genome that you are trying to construct. So a reference genome helps you to assemble a new genome, also from short reads especially. And we can see from this illustration how you have your short reads coming together, being guided by a reference so that you can anchor those short reads on the reference, helping you to form a contiguous assembly or scaffolds, as the case may be, since you may have gaps within the assembly. And the reference assembly also helps as a coordinate system for reference, just like the name implies. And as a coordinate system, what it does is it gives you the specific loci where you may have features of interest based on whatever analysis you have carried out. For instance, here we have a kind of an address that tells you about a feature on chromosome 7 at position 348729 to 348779. That's about 50 base pairs. And also another address or another locus at chromosome 12, 8374, etc. Now, with this coordinate system, everyone making analysis about a particular species can use it as a reference, and then anyone that wants to make comparison or to confirm can go back and pinpoint the specific address. So in all this, reference genomes or a single reference assembly has proven very useful up until now. It is also important to know that many functional genomics assays that we have to help us understand how genes perform their functions are talking about assays like RNA-seq that helps you to quantify transcripts, or even assays that help you to determine or discover variants in your assembly. They've been based on short reads. And because of that, you make use of a reference genome, a single reference genome, to carry out the analysis that you want to perform. Now, I want to quickly highlight these limitations that we have to a single reference with this illustration. Let's assume that we have this reference assembly, and on this reference assembly, we have two loci, about 1.5 kilobases, and then another loci, another locus, about 1 kb, that's 1,000 base pairs. And we have two other assemblies, another assembly B, where you have some variants, but on this particular assembly, you have the regions that are absent on the reference. That's the regions that are absent on the loci L1 and L2. You have them present on assembly B, and some of it also on assembly C. Now, for assemblies B and C, they represent individuals that carry some true structural variants within them. And these structural variants have been highlighted in these colors, where these structural variants range from your insertions, deletions, or even your single nucleotide polymorphisms. Now, the current pipelines that are used for variance discovery would take short reads and then align the short reads to the reference assembly. What the pipeline would do is any read that does not align to the reference assembly is discarded as artifacts. Now, in a case where you have true variants in these individuals, B and C, and these variants have been linked to some very important traits in these individuals, perhaps heat or drought resistance, or even disease tolerance. And you want to determine these variants, you want to find out where these variants are located. With the use of a single reference, you are not going to find those variants because the pipeline will simply discard these short reads because they do not align to the reference genome. That is one of the problems of reference bias. Now, another problem which is not illustrated in this panel, in this panel is that of erroneous variants, which is due to short reads aligning to multiple regions of the genome, which is different from where they originate from. So in this scenario, you have a very likelihood of short reads when you're aligning to a very large genome. You have a very high likelihood of these reads aligning to multiple places all over the genome. And because of that, you could determine or you could discover false variants in loci where they do not have origin. And all this compound the problem of variant discovery based on a single reference genome. Now, if you want to start looking beyond the use of a single reference genome, how do we go about it? Now, the first case or the first instance of talking about multiple reference, as it were, instead of a single reference, came from this study from Tettelin in 2005. That was almost 2 decades ago. And they were analyzing different strains of the bacteria, Streptococcus agalactiae. They used 8 strains to be specific. And what they found out was this illustration of the different parts of these different strains, the genome of these different strains that are different. It was so profound, and it was the first time that such a phenomenon was being observed. And they tried to see if there was a way to represent this large diversity that they have in this genome. And of course, at that point, it was not possible because the data structures that are being used currently, they had not been developed to that point to adapt it to the use of multiple genomes. Now, they made note of this and also tried to estimate how many individuals in this particular species would be sufficient for us to be able to represent the diversity that we see. For that particular one, it was difficult for them to estimate because there's something we talk about in terms of estimating the correctness, the completeness of a pangenome, and that is whether the pangenome is open or closed. So they discovered in this case that this particular pangenome is open. Now, the complete genome length of this strain of the bacteria is just about 2.1 megabases. That's just 2 million bases. And for some context, when we look at this size compared to the size of a cattle genome, the cattle genome is about 1,400 times the size of this particular gene. And this particular gene, this particular organism has just about 2,000 genes, whereas the cattle has over 40,000 genes. So when you look at all this, with the technology available at this time, it was very possible for them to get a complete genome and make a pairwise comparison, a multiple comparison between all these strains. But for species like the cattle, it was difficult because we needed more advanced technologies to be able to get the scale, chromosome assembly scale genomes that would enable us to carry out studies like this. Now, scientists knew about this problem. They knew about the challenge of the single reference genome, but the technologies available at that time did not permit investigating species at pangenome scale. Now, in the last 3-4 years or thereabouts, we've experienced some advances in the sequencing technologies and also genome assembly technologies that enable us to assemble a genome completely without the use of any reference genome, and that is doing a de novo assembly. Now, these technologies that enable this include the use of high fidelity reads from PacBio, which has tremendously increased how many bases we're able to sequence at once compared to the first and second generation sequencing technologies that were in use. So with high-fi reads, we could get a single read that is up to 20,000 bases, that's 20 kilo bases long, and at very high accuracy, up to 99% accuracy. And we also have ultra-long reads from Oxford Nanopore, which enables us to have a single read that spans up to 100,000 base pairs, that's 100 kb, although the accuracy, the base accuracy at this length is reduced compared to what we have for iFile reads. So new technologies, new assembly technologies exploit the strengths and weakness of both technologies to be able to assign reads correctly to genomes. And with this, we are able to get more complete, more chromosome scale complete genome assemblies of different species. Furthermore, the trio binning approach, which is being used successfully to help to face haplotypes, has been instrumental in getting truly and completely phased haplotypes of different individuals. How does this work? So for a diploid individual or for a diploid species like a cattle, you have two copies of the genome in an individual. So one copy comes from the sire and the other copy comes from the dam. As we have on this panel, here we have the sire with the copies of the genome colored in blue. Then you have the dam on the other side with the copies colored in red. So the cross that comes from these two individuals will carry a copy of the blue and another copy of the red genome. Now, what trio binning does is, using short reads, it tries to get markers. It tries to get short sequences that are unique to either parents. So the short reads of either parents are used to identify markers that are unique to those parents. And then from the long reads of the child, from the long reads, then you're able to separate the sequences that belong to the copy of the genome from the sire, from the sequences that belong to the copy of the genome from the dam. And this is what is represented with this Venn diagram here, where you have there's a red circle for the dam and the blue circle for the sire. So what you're looking out for in the specific markers are those parts colored in blue that would help you to identify and segment the reads into the parts that are from the sire and also the parts that are from the dam, as we see on this illustration. So with this, we can see that all the reads that we have on this side have blue markers that have been identified using those short sequences that were specific to the sire. And all these reads, these long reads on the other side, belong to the dam because of the red markers that are here. So the assembly of the two genomes are then completed with only the sequences that are from the dam and also the sequences that are from the sire. So this is what we call haplotype phasing, that enables us to be able to get only the sequences that are from a particular parent of an offspring. Now, this technique has proven very useful, especially when you have crossbred individuals, helps you to retrieve completely and truly the complete genome of either of the parents. And this recipe is currently being used in so many of the assemblies that are being generated currently. Then the fourth technology that makes this possible for us to get complete de novo assemblies is high-seq technology. So high-seq technology gives us information about true interactions between chromosomes in the cell, helping us to be able to assign the chromosomes completely, the contigs that come from the chromosomes completely, and phase them. Phasing is to determine what haplotype it comes from, and also help us to get the correct orientation of the assembly. Now, with all these new advances in technology, it is very clear and evident that any variant calling attempt that we may want to embark on would only be as good as the quality of the genome that we want to use to call variants, and also the reference that is being used. Now, for a pangenome is regarded as a set of genomes. And this set of genomes, of course, comprises the genes and other non-regulatory components that we have in the genome. And comparing or bringing together all these genomes in this set, we have three major components of the pangenome that are worth considering. Now, the first component is the core component. And what the core component comprises of are those segments of the genome that are common to all the individuals that are used in constructing the pangenome. So, for instance, in the core, you would find a lot of housekeeping genes or a lot of regions that are responsible for housekeeping, that is, for keeping the organism alive, for keeping the species alive. And because of that, it is common to all the breeds or all the individuals that you have in that species. Then you also have another segment where you have traits or where you have genetic component that is shared between one or more individuals in that pangenome. And with this region, which is called the shell, you have a lot of variation coming up from this region. And it helps us to be able to interrogate these different individuals and help us to understand the evolutionary relationships that we may have within this breed. I mean, the different individuals in the breed or the different breeds in a species helps us to understand the evolutionary relationships, and then whatever pressures, whatever evolutionary pressures that these different individuals or these different breeds are responding to. So this region, which is the shell, holds a lot of information, holds a lot of useful information that helps us to understand more about the species, either as breeds or as individuals. Now, the third component of a pangenome that we have is the cloud. And what this cloud component is made up of are those specific or those unique traits to each individual that make up the pangenome or to each breed in a species. Now, this region appears to be a gold mine of some sort that would help us to understand whatever traits are responsible for, whatever genetic drivers, whatever variants are responsible for some traits that are observed in a particular individual or a particular breed that may be useful in another breed. Now, for instance, we know that the genetic component that controls coldness in an animal is just a 212 base pair insertion that represents or that displaces a 10 base pair sequence on the carton chromosome 1. If, for instance, we're interested in transferring this trait into another animal that is horned, then we know where exactly to look out for and introduce this particular genetic driver into the new animal, instead of relying on doing multiple crosses that could even result in losing some traits that are beneficial in the target animal. So this helps us to narrow down and do a more precise breeding for the specific traits that we are interested in. So with pangenomes, we have an opportunity of mining this information source to help us to specifically breed for the traits that we are interested in. Now, pangenomes are represented as what we call variation graphs. And a variation graph just helps us to capture the diversity, to capture the differences and the common regions that we have in the assemblies that make up the pangenome. So for instance, on this panel, we have all these solid blocks represent the regions that are the same, that are common to these 10 different genomes that we have represented. And we have other parts that vary, that are common to some of them, that are missing in other genomes. And then we have some that are just single nucleotides, and these are the SNPs. And then you have others that are longer, either as insertions in some genomes and deletions in others. Now, trying to represent that or trying to visualize that even better, we see how to move around or how to traverse a variation graph to be able to recover or to be able to get the genome of interest. For instance, at this junction, we have a movement from the third nucleotide to the seventh one here, because there is a deletion here that is not captured in the variation graph that will represent this region. And for an insertion, it only takes you, it traverses from the edge of this nucleotide, connects the nodes, and then move on to the next node. And for a single nucleotide variant that we have, you have two variants represented, and you could traverse in either path. You traverse the genome either path to recover your genome that carries that particular variant, leaving out the other one. Now, the other variant that we have is when you have a copy number variation. Looking at this particular region, we have a tandem repeat of these three nucleotides, TTC, repeated 3 times. And for this traversal, which jumps from the first copy to the third copy, it means that a genome that traverses this path has just 2 copies, whereas a genome that will traverse the other parts going through the 3 copies have 3 copies. So with this structure, it is easy to represent all the different kinds of variation, all the different kinds of variants that we can have in a particular genome. For SNPs, for instance, we have been able to use SNPs well with respect to the current analysis techniques and the current pipelines that are available compared to the other variant types, that is indels and copy number variants. Because the tools that we have currently for all this, they've not been able to do such a good job that we have done on SNPs. So the exploration of the contribution of indels and copy number variants to understanding traits have been limited compared to what we have obtained on our SNPs. And so using a pangenome would help us to capture all these types of variants, all these other types of variants that have been difficult to explore with current tools prior to now. So in order to also understand better how a set of genomes are collapsed into a single graph, or otherwise how you retrieve a set of genomes from a variation graph, this small illustration, which gives you the graph as a set of nodes, and then the edges that connect the nodes represent the relationship between the nodes. So here we have a total of 6 nodes, and from here we can retrieve 3 different genomes by traversing different paths. So we would see that in the first path, you have the first big node moving over to the next, the smaller node, the first small node, moving over to the third node, and then the fourth one. This traversal keeps this third node because it is not present in that genome. That's why it's absent here. Then the second genome traverses the 2 nodes. It connects the third node, the fourth and the fifth nodes. That is why you have all these copies present in this genome, while the third node, the third genome traverses only 3 nodes, which is represented by the 3 blocks that we have on here. Now with this, it is important to state that the construction of a pangenome, or as it were, the usefulness of a pangenome, is dependent on having very accurate and complete genome assemblies. Now, what this does for us is it makes a case also for getting what we call a telomere to telomere chromosome level assemblies that is complete and is able to represent all the features that we have in our chromosomes. For instance, we know that all the chromosomes that we have on cattle are acrocentric except for the sex chromosomes. So it's important that we have assemblies that is able to read through the heterochromatic regions and also the edges, the telomeric edges that we have on these acrocentric chromosomes. Now, with the fact that we're able to construct genomes de novo, it is also important that we are able to assess the quality and completeness of these genomes without any recourse to a single reference. And that is why we have these days, we have tools that enable us to be able to assess how complete our assemblies are without making any reference to a single reference genome. And that way, we independently know whether we have a very high quality genome. Then we're able to determine whether we have a complete genome based on how many of the chromosomes in the genome do we have to be telomere to telomere, as it were. That is, observing telomeres at the ends of the chromosome where they have telomeres. With all this, we are able to construct very high quality genomes that lead only to high quality new reference, because a pangenome constructed from very high quality genomes becomes a new reference in lieu of a single reference assembly. Now, some studies have carried out pangenome analysis on cattle, and they've shown that pangenomes compared to a single reference genome make a lot of difference. Now, in this study by Crisanto, they compared or brought together 5 different species, 5 different breeds of cattle, constructing a pangenome from 5 different breeds of cattle. And from these, they were able to recover about 70 million base pairs that were not part of the current, of the ARS 1.2 genome. We have a newer one now. So out of this 70 million base pairs, we would see that only one breed contributed so much to the number of base pairs that we have recovered that did not belong to the reference assembly. Now, when we look at this plot here, we will see that we have quite a huge representation of the shell. If you remember, when I showed the different regions of the pangenome, where we had the core, the shell, and the cloud, you would see that we have quite a lot of this represented in the shell. And then the cloud, we see a lot of variants too, also from the cloud. And from a single individual, which is the yak, it accounts for the most of the new base pairs that were added to the pangenome that were non-reference. Now, what they did further was to try and find out how much of this region belonged to the coding regions of these assemblies. Now, doing RNA-seq analysis on all these assemblies, we're able to identify almost 1,500 genes that were not in the reference assembly. And out of all these, 885 of them were expressed. So the implication of this for previous studies using a single reference is all the genes that are in this region that are responsible for some of the traits that have been identified in some of these breeds, all these genes have been missed prior to now. But it is only when a pangenome was constructed that this was revealed. And with all this that has been lost, a repertoire of very important knowledge has been missed for so long, but the use of pangenomes can make us to begin to interrogate and exploit all this useful information that we get from the use of a pangenome. In another study by Leonard et al., they also constructed pangenomes to identify the variants that are responsible for some traits in some species. Now, 3 different genes especially were identified that were responsible for some traits in this particular work. And in the first one, which is what I'm hovering over here, we have the Q-Rich2 gene, where they found variants based on a 30 base pair tandem repeat. And this particular repeat was found on the 5th exon of this particular gene. And what is this gene responsible for? The gene is responsible for sperm mobility. So the implication of this is this variation that we have here would affect the fertility of male cattle. And what this does is, if we have variants that is responsible for improved fertility, then we can get the information that, the genetic information that improves this and incorporate it into other species to also improve their fertility, especially if that locus is missing from that species. Then the second gene that they identified with another variation is the TAS2R46 gene. And the genetic variation that was discovered in this was a 17 kb deletion. And what this gene is responsible for is beta taste receptor that contributes to the dietary adaptation of the animal. And looking at this, we see that looking at this, we have 2 tracks of genome, one going up here, and the one that goes up here also has different variants, different combination of variants. And these are the things that were identified, these are the regions that were identified on this particular gene that is responsible for this dietary adaptation in the animal. And the third gene that was identified, which is responsible for meiotic recombination, is PRDM9 gene. And this variation that was identified here is also a copy number variation of the zinc finger proteins, where you have different copies of the tandem repeat of this zinc finger protein was responsible for controlling the meiotic recombination in cattle. Now, these studies point to, or they give us a glimpse of the value that we could get, that we could obtain from using a pangenome over a single reference assembly, especially when we are calling variants. So another difference or another advantage that we derive from using a pangenome, it comes from the use of low pass or low sequencing, or low coverage sequencing, or low pass whole genome sequencing. And what does this mean? With low pass or shallow sequencing, we could just sequence genome to maybe 1 or just 2x depth. And what this means for us is we'll be sequencing at a much cheaper price or much cheaper rate, but it is reliant on ensuring that we have a very accurate and complete genome that makes the combination of a low pass sequencing and variant calling very possible. Now, with low pass sequencing, we easily know that the reference to which we are using is high quality. The likelihood of getting erroneous variants is very reduced. And this gives us much accurate results compared to deep sequencing with a single reference assembly for variant calling. Then for producers, the use of pangenomes would enable us to understand or to explore how structural variants, not only SNPs, other structural variants like indels and copy number variants, how these are connected to the traits that we observe in our livestock species. And the importance of that cannot be overemphasized when we are doing selective breeding. When we are interested in some traits that are controlled by variants that we are able to accurately detect, then it's easier for us to incorporate those genetic components to be able to get the same traits in our species of interest. Now, it also helps us to make better genomic prediction because we're working with more accurate data, we're working with more complete data, where you don't have missing variants in codes just because they are not present on the reference that is being used in the case of using a single reference sequence. Then the use of pangenomes help us to exploit the genetic diversity that we have in breeds, just like I had mentioned, looking at that cloud region of our pangenome that helps us to identify those specific traits that will be helpful, that will be useful, especially the ones that are economically beneficial to our livestock. So I'll briefly introduce the bovine pangenome consortium and what has been done so far concerning this project. So the bovine pangenome consortium aims to, as much as possible, capture the genetic diversity that we have in cattle across the world. It's a global collaborative effort with scientists from different parts of the world, and different cattle species from the different breeds are going to be sourced, and these are going to be analyzed to produce pangenomes that would capture the different variation that we have in each of these different species. So looking at this phylogenetic tree of cattle, we would see that we have different breeds that are colored differently, and the colors represent the different parts of the world where these species are found. So we'll see that we have some in green. These are the species that are known to come from Asia. The red species are from North and South America. The ones in black are from Africa, while there is one here that we have in orange, which is from Australia. All the blue species are from Europe. So with this, you would see that we have a broad representation of the different species, which are the ones that are highlighted with the black asterisk or the black star symbol, a broad representation that helps us to capture whatever variation that we may have across the world to a very large extent. And currently, work is ongoing at different stages, at different levels to be able to carry out this mandate. And right now we have at different stages of either sample sequencing or assembly, up to 18 different breeds that have, in some cases, we have multiple individuals from a particular breed. And all these are still work in progress. But very soon, a data freeze will be imposed on this when the assemblies are done, so that initial analysis will be carried out across the spectrum of all the breeds that have been collected so far. Although some of these are still being, the samples are still being sourced, which is constituting a bottleneck, especially for the ones that are in places where the local capacity to do the sequencing may not be there. Otherwise, sequencing could be done locally, and then the data will be analyzed. So all these breeds have, to a very large extent, represented the different parts of the world where we have the different breeds of cattle. So This slide is to give us an insight, a quick insight into some of the things we have learned from this bovine pangenome project, which is based on 2 species whose Y chromosomes are being currently analyzed. I remember I told you that some samples are still being sequenced, and because of that, a breed-wide comparative analysis is not yet possible. But for some of the breeds we've been looking at, especially for the Y chromosome, we have 2 species here, a Wagyu beef cattle, and then you have an Ayrshire dairy cattle. So the differences that we've noticed between these 2 particular breeds are in the structure and then the length of the Y chromosomes. The Y chromosome is the critical chromosome that determines the sex of a species. And in this case, for the Wagyu, we have a shorter Y chromosome compared to what we found on the Ayrshire. Now, I'm showing this structure just for us to see that between the 2 Y chromosomes, we have a similar structure. And then the lengths are different. The structures are similar based on this marker that is shown here, which is a repeat sequence. And this region, where you have a concentration of this repeat sequence on both chromosomes, are showing where we have the centromere located. So we have a centromere about the same length from the P end of the chromosome, but then the distribution of the rest of this repeat structure is similar in both chromosomes. Now, what this tells us is there is a structural variation somewhere that accounts for this disparity in the length of the 2 chromosomes, despite having the same kind of structure that is based on this marker that is shown on this slide. Now, in summary, what the use of pangenomes, what it tells us, or why we are using pangenomes, is the fact that the use of a single reference appears to be insufficient to capture the diversity that we have in different species. And the advancements that we have in technologies have enabled us to be able to explore this at a larger scale with the use of hi-fi sequencing, ultra-longs, that enables us to generate complete and accurate genome assemblies, chromosome scale genome assemblies. And the use of this set of high-quality assemblies would enable us to generate that high-quality reference pangenome that would capture as much as possible the different variation that we have present in the species or in the individuals that make up the pangenome set. And then with this pangenome, we're able to link the genetic variation to the traits that we have that are observed in the different breeds of this cattle species. And the Bovine Pangenome Consortium is currently making concerted efforts to get as many species that will represent the variation that are present in the different cattle breeds that we have across the world, in order to construct that pangenome that would be high quality reference, that is useful for variant call and helping us to understand traits and for better breeding. So with this, I would like to acknowledge my supervisors for their guide and their help at all times, and also thanking the Beef Improvement Federation for giving us this opportunity to share these thoughts. And I would like to acknowledge also the Bovine Pangenome Consortium and the Ruminant Utility Consortium. Thank you for your attention.